Okay, we're ready. Everybody's good. Okay, so my name is Scott Bowdridge. Just to give you a little background about myself, I grew up on a sheep farm in Southern California, in between Los Angeles and Las Vegas, out in the middle of the desert. My family raised Rambouillet sheep. Um, we showed sheep all over the western United States. I went to college in Chico State in Northern California, judged livestock for that small college there. We're pretty competitive nationally. And then I was a high school ag teacher in Central California. Right? We forget sometimes that California is the #1 ag state in the country. But I did that for a little while, and I moved to Maine. And I ran a nonprofit rare breeds farm for the guy who invented the Ethernet. And him and his wife ran this nonprofit rare breeds farm. So we had Cotswold sheep, and I was doing that. And while I was there, I met some guys that were doing research with Katahdins. And that's when the Katahdin project started. So this was around 2000, about 20 years ago. Creeps me out to say that out loud. So I was in Maine working with Katahdin sheep, and I did my master's degree at the University of Maine looking at some different genes in Katahdin sheep. I fancied myself as a geneticist. So any good sheep geneticist would want to go to Virginia Tech and work with Dr. Notter, who helped develop EPDs in sheep and EBVs and doing that stuff. So I did that with Dr. Notter. He told me in the hallway that I was not a good geneticist, but I was a better molecular biologist. So I am a trained immunologist, right? So I did my PhD at Virginia Tech, and then I went to do a postdoc at a medical school in New Jersey before my wife and I moved to West Virginia. So I've been at West Virginia for about 10 years now, and we exclusively study parasite immunology in sheep. And I use St. Croix sheep as the model for parasite resistance in sheep. And that's what we're going to talk about today. So this talk, I wasn't quite sure what the audience was going to be like and how everything's going to work. So it might be a little denser than what you expected. So just go with me. There's going to be a lot of terminology and a lot of things like this, but you'll understand the basic principle that we're trying to get our arms wrapped around is how does it work, right? So we're all mechanics trying to pull this engine apart with no idea of how the engine works. So we're just trying to take it apart piece by piece and understand, okay, how does this piece fit in to how the engine operates? So that's what we're doing with this immune function. So let's go to our next slide. So just to give you an idea, if you look at some of the sheep statistics in the U.S., we lose about 35,000 sheep a year. Of those 35,000 sheep that we're losing a year, those are death loss due to parasites. So that accounts for about $700 million per year and about 12.5% of our industry's total value, right? So that's what we're losing to parasites. They are the #1 reported disease. So after predation, the next most reported incident in sheep is parasitism. So go to your next thing. So understanding how the life cycle works, and we're only talking about one parasite, and the parasite is the barber pole worm, and it's called Haemonchus contortus. You guys have heard this multiple times at multiple meetings, so we're going to do this again. I've got really cool animations to do all of this stuff. If we start with what's coming out of the south end of that sheep, and what's coming out in the poop over there, we've got those eggs. And the eggs are generated by adults that are mating inside the most inhospitable place in the digestive tract, which is the abomasum, which is just like our stomach. So it's a highly acidic environment. The parasites shouldn't be able to live there, but they do. They love it there. That's their niche. That's where they do that stuff. So they're going to mate, produce eggs. Those eggs are going to be shed out into the pasture and into the environment. So in the environment level, so below the dotted line, you're going to have different larval stages of development. So it's going to go through an L1, 2, and then finally at its L3 stage, or that's the stage where it's infective. So when sheep consume that consumed L3 is going to migrate with all the food that it's eating. It's going to go in their rumen reticulum, go through the omasum. It's going to get through the abomasum and stop. And then those larvae are going to start developing into adults, start mating, and the process starts all over again. So from the time of ingestion of an L3 larvae all the way to the production of eggs is anywhere from 14 to 21 days. The time from those eggs to become infective stage larvae in a good environment that's nice, warm, hot, humid is about 7 days. Can it happen as early as 3 days? You bet. So that can happen when it's midsummer, and Haemonchus really likes it when it is hot. When it's hot outside, when it doesn't cool off much in the overnights, when it actually is, sometimes it's more humid, like at 5, 6, 7 o'clock than it is in the middle of the day, it really loves that. Go ahead and click the next thing. So this is what it looks like on the inside of the abomasum, and you guys can't see it in the back because it's small, but that little guy right there is your parasite, this one right here, this one over here, this one up here, and they don't attach. It's not like a hookworm. They have no features around their head that allow them to attach, so they just have to swim to kind of stay in place. They produce a lancet at the end of their mouth, and they poke the abomasum, and they open their mouth, and the blood just goes in, right? And that's how they get it. So we call it a barber pole worm because its intestines are wrapped around their ovaries on the females, and it makes it look like a candy cane or a barber pole, right? So that's how they get their name. Go ahead and next one. So ultimately, if this is left untreated, you guys know what happens. I've dealt with producers all over the country in dealing with this issue. It's not something that is located here. It's not something that's just exclusive to the southeastern United States or even in the Northeast. I've dealt with producers in South Dakota, Wyoming, California that are having problems with parasites. So the thing about in California, we don't get any rain, right? So we have to rely on irrigation. Well, we're pumping water out on pastures and keeping them warm, moist, and humid ourselves all the time. So we're getting problems in irrigated pasture lands. And I think that's a lot of this research moving forward is going to be looking at that in western states that use some more irrigated pasture lands. Go to your next slide. So if we think about what makes an animal parasite resistant, right? So what we're going to talk about today is this first thing. There is a high immune response that prevents larval establishment. Go ahead and click down. That does that. So if I don't get larval establishment, I don't get any adults, I don't get any eggs, then I get no pasture infectivity, I don't get any host death, and I don't get any other animals infected. That's what parasite resistance looks like. Go to your next slide. So we can see this in St. Croix sheep. So I started using St. Croix when I went to Virginia Tech in 2006. And then when I started at West Virginia, we were able to keep those animals completely free of parasites their entire life. So they were born in a barn, in an elevated floor barn. Everything was there. They weren't exposed to anything, and we could do this kind of work. So what we would do is we would take those lambs that were born, we'd keep them out on that elevated floor, wean them, wait 3 or 4 weeks after weaning, make sure they get through all that garbage around weaning, and then we would give them 10,000 larvae, just orally. So we've got some other sheep that we keep around that we keep persistently infected to generate larvae for all of our studies. So we would give them 1 dose of 10,000 larvae. And what would happen, and that's our primary infection group. There's that red line. So it goes up to about 3,500 eggs per gram, and then the next week it drops out. I didn't deworm them. I didn't do anything. Nothing. Right? And remember, they're not grazing out on pasture, so they're not getting reinfected on a daily basis. This is one time of infection. They get up to 3,500 and then go away. Then what we did is, just for safety, we deworm everybody, just make sure they're completely clear. We wait 2 or 3 weeks. Then we come back and we give them another infection. That's the challenge infection. And then look at your green line on the bottom. So when I come back and do that again, that's challenge. We have all become armchair immunologists living through COVID, right? We're all familiar with antibodies. I heard somebody on TV talking about memory B cells the other day. It's crazy. That's immunity. The ability to clear that infection without it becoming patent or becoming active is effective immunity. Go to your next slide. So if we think about animals that are susceptible, so I get a lot of garbage from the Suffolk folks because they don't like me sometimes, but Suffolk sheep like to find a way to kill themselves. In this case, they don't generate an immune response. You can click down. And by not generating, go ahead and click down. By not generating an immune response, you allow larvae to establish. The adults start mating, they produce eggs, you get pasture infection, more parasitism, and ultimately more infective level in the animals. So to see this from a graphical standpoint, next slide, we can see that what happens in the Suffolk crossbred lambs During that primary challenge, I didn't give them any more larvae than I did the Croix, but their fecal A count goes twice as high as the Croix do. They never clear it, so it drops down right there. I have to explain this every time because I'm not going to repeat this experiment. We had the guys feeding that week, just threw a bale of hay in the pen. Everybody got diarrhea, and diarrhea dilutes fecal A counts. So it goes down and then comes right back up. Okay, so that's what happens right there. It's nothing. There's no immunity or anything that's happening, but that's just diarrhea. So during the primary infection, they stay highly infected. Then when we deworm them, rest them for 3 weeks, and come back and give them another infection, they still get infected. So Suffolk sheep have to see this over and over and over. So think of it like boostering for this COVID vaccine. Everybody's talking about this now, right? The more times you see it, the better you get at doing it. But some animals are just better at it immediately, right? And some breeds are just better at that. And we've noticed even the scientific literature about COVID, some people are more resistant to COVID naturally. Okay, it's just hard finding those people, right? They're difficult to identify. In this situation, we can see that really clearly between those two breeds. So there's a real obvious difference between them. So then our job is to know, okay, so that's the engine. Now we have to start taking it apart and finding how all those pieces fit together to do that. So go to your next slide. So what we started doing is we would start killing animals after infection. And when we did this, we were able to take pieces of the abomasal tissue. And this looks like it doesn't make any sense to anybody. You don't know anything yet. So I'm going to step away from the, actually, I'll just take this with me. And then that way you guys can hear me still. So in the first one is an uninfected animal right here. So there's no, all these little purple things, those are cells that are in the lining of the abomasum. Okay, so that's, and the abomasum, if you open it up, there's folds inside of it. So all we did is we took a cross section of one of those folds, we stain it, you can look at it at a microscope, and then I had to spend my college career counting these little purple dots, right? So that's what I did in school. This is the just an uninfected animal. Then this one in here is B. This is a Suffolk sheep. This is from a Suffolk sheep that had been infected for 7 days. So there's some cells we see right out here in this area. Oh, you're going to move the camera. There you go. Some cells you see out in this area, but not a whole lot. This is what most of the St. Croix sheep look like. And see all of these purple dots everywhere? All in the middle here. So this light pink area is the muscle lining of the abomasum on the inside of it. All those cells are infiltrating in and they're coming out into tissue. So you're getting a cellular immune response, and then we can quantify how many globule leukocytes and eosinophils. But the interesting thing is that there is a difference in neutrophils, and neutrophils are kind of our first responder cells. That's what they do. That's what they're the first ones to get there, and the Croix are responding quicker. And that's going to be the moral of this story over and over and over again. You're just going to hear that Croix respond quicker. Go to your next slide. Okay, so then we started counting lymph nodes. You guys know when you get sick, the doctor goes in there and starts feeling lymph nodes, right? So you have lymph nodes inside your stomach and inside your gastrointestinal tract. Right next to the abomasum, there are lymph nodes there too. So I can count them, I can weigh them, I can cut them in half and measure the area just to give another quantification of how big they are. The St. Croix sheep are responding. So there's this black line. So this is uninfected, 3, 5, 7 days after infection, we're getting just this increase in lymph node weight. It's not happening in the wool sheep in this example. And these wool sheep here that we used in this experiment were Rambouillet, Dorset, and Fen sheep crossbreds. So they're not getting it done in this case. And then we can see lymph node area is going up. So it's not the function of how many lymph nodes are there. It is the fact that the lymph nodes are themselves getting bigger. And that tells you that they're generating an immune response. Next slide. Okay, then we can do all of this. So now we're going to take and venture into this crazy immunology area. So this is where it gets, this is like pulling apart the transmission and trying to put all those little rings together and getting them all aligned and everything else. So we've got this little chemical here that recruits neutrophils. Well, by day 7, it is higher than it is in the wool sheep. This one recruits another type of cell. It is higher in the St. Croix sheep than it is in the wool sheep. This one, this is the defining cytokine or our immune response for parasites. It is present in the Croix and not present in these sheep, period. Okay, and this is just, we're looking at gene expression. We're looking at these genes being expressed. And this is another one that looks at all these. That's another part of this kind of cytokine group. Cytokines are just hormones of the immune system. So it's nothing different than you guys putting cedars into animals, those hormones that are involved in that. These are just hormones for the immune system. So we're getting IL-5 differences. So that's, you get this early, rapid, multi-pronged response from St. Croix sheep to clear out larvae. That's what they're meant to do. Next slide. Okay, so we stopped at day 7, right? And most of the literature stops at either day 7 and then it skips to day 14 when those worms start becoming active. We decided to kind of go in no man's land, right, and look at day 10. Well, what happens at day 10 here, and these are Suffolk sheep at this point, they're all infected with larvae, and this is a challenge infection, so we found on average 16 worms in the stomach of the hair sheep and 530 in the stomachs of the wool sheep. And this would be Suffolk sheep at this point. Then we looked at the weight. Well, the weight between the hair and the wool at this point are about the same, which was confusing us because we expected the hair sheep to still be higher. What happens is that the hair sheep, what they're at right here, they're going down from where they were 3 days ago. So at day 7, they were actually higher, and day 10, they're starting to shrink. The Suffolk sheep are just catching up. They're just starting to respond. And here, remember I showed you the IL-4 stuff earlier before? It was there, and it was not there for the, now it's reversed. And it's there for the Suffolk sheep, but not there for the St. Croix sheep. So what does that mean? Next slide. So we get reduced worm burden in St. Croix sheep. We get lymph node growth by day 7 in St. Croix sheep. We get IL-4 expression in St. Croix sheep. And we get neutrophils in St. Croix sheep. This shows us that these sheep respond rapidly to this. That's what makes them parasite resistant, is that they respond immediately to this infection. And they don't wait around for something else to happen. The longer you wait, the more opportunity there is for damage to occur. So they're going to start bleeding, they're going to start taking blood meals, there's going to be more. Let's just think about an animal that goes out on a pasture. If they graze once and pick up those worms and then don't graze again, they might be able to handle it. But these sheep on a daily basis are bringing in parasites. So they're doing this. So if they have a delay in their response, you'd have a delay and then boom, a big level of infection in the animals. Next slide. Okay, so all of this comes down to these immune responses. So if you've got this worm coming in here, you've got eosinophils and neutrophils. We wrote another paper about how neutrophils can actually spit out their DNA and then will wrap up the parasite. And by wrapping up the parasite with their DNA, the parasite can't swim. And if it can't swim, it can't hold position, and it goes out with everything else. So that's one of those mechanisms that might be used by the immune system in these sheep, and then that mechanism does not happen in the Suffolk sheep. So those are some interesting things that happen with that. But then we get all of this other stuff that's going on and trying to understand, this is the engine, right? And these are all of our little pieces. So we have to start pulling each one of these apart and taking a look at them. Go to your next slide. Okay. So without completely boring everybody with all of the nerdy immunology and doing all that stuff, I wanted to share some of the work that we did with Katahdin sheep, just to flex our muscles a little bit and showed it. We know that St. Croix influenced the development of the Katahdin breed. We know that Katahdins are the #1 breed in the United States. There's more annual registrations for Katahdin sheep than there are for any other breed. The next two breeds combined still are just close to Katahdins at that point. So it's scary when it used to be Suffolks dominated everything and everything else was behind, and that whole paradigm is flipped now. So our working Katahdin sheep, go ahead and click down. We started working with, so we partner a lot with Virginia Tech, and there's a flock of sheep, or flock of Katahdin ewes down there that we had a large USDA grant to start looking at the impact of selection for fecal A count breeding values. So we took rams that had very low fecal A count breeding values, so they're negative 99 for fecal A count breeding value. Then we had rams that when we bought them were like plus 1,800 and actually had progeny on the ground and had accuracy and data with them and everything else. They got adjusted back to 666, so we called them the devil ram. So we had that one, and we had our minus 99 rams, and we mated those to a random group of ewes, right? Just throw those low boys, you get this group. High boys, you get this group. This first year that we did this study, we started looking at death loss in the lamb crop. The black bars are the ones that were sired by low EBV rams. 10% death loss. You guys okay with the 10% death loss in your lamb crop? No, this was a bad year for everybody. We had an outbreak of Clostridium type A that we don't even vaccinate. Nobody really vaccinates for. But in that environment where there was a lot of Clostridium A, if you were on the high side of that fecal A count, EBV, you lost almost 30% of your lamb crop. This group, we only lost 10%. Okay? The next year we did it, we actually took low rams and bred them to low ewes, so they were less than 0 for their breeding value. High rams to high ewes. The guy that runs the station down there just looked at us like we're crazy. He's like, all these sheep are going to die. Why would you do this to me? And I said, well, just do it for 1 year. We've been doing it every year since to expand on all this stuff. And we started looking at some other things. Go ahead and click to your next thing. Of the percent of ewes lambing, this is the number of ewes with mastitis that were culled based on mastitis. If you have low fecal A count breeding values, your chance of being culled, you're about 2.2% of those ewes were culled. About 12.5% of those ewes were culled because of mastitis on the high side. So what this tells us, or at least gives us an inkling of, is that when we select for fecal A count, or we select for parasite resistance, we're also selecting for improved immune responses. Okay, go to your next slide. So to prove that and kind of identify, we did this whole experiment, right? We had these low rams that were mated to low ewes, high rams that were mated to high ewes, and then all the lambs were managed together. When I talked about that death loss excluded any lambs that died or had any parasite problems. So this is everything else but parasites. We really didn't lose any lambs to parasites at that point. It was everything. It was pneumonia, coccidia, those type of things. Go to your next slide. So we would take blood samples around the time that we vaccinated the lambs. So we're trying to see if the low sheep or the low EBV Katahdins generate more immunity to a simple thing like a vaccination. So this would be like after your COVID shot. If we got your COVID shot right here, and then you're waiting 2 to 3 weeks to get your booster, right? Same thing here. We're going to take that first blood sample, and then we're going to take 3 more blood samples. You're going to get a booster, and then we're going to take 4 more samples after your booster so we can see what your antibody response is to the vaccine. Go to your next slide. So when we do that, what we get is that here's just fecal A counts, right? Guess what? If you select for parasite resistance, they are lower on their fecal A count breeding value, or they're lower on their actual fecal A counts. So these guys are around 500. These ones are up over 1,000. You can see the difference in their fecal A counts as we go over these 8 weeks. Next slide. Then we will start looking at antibodies. So in the first year we did this, the low group right here just tend to be a little bit higher all the time, right? Just a little bit higher. And this first year, we only used the sire selection. So we only used low sires, and we randomly mated them to ewes. And then high sires mated to another random group of ewes. We just get this little increase. But then the next year, when we look at it in the lambs that come from low ewes and low rams, go ahead, next slide. It's all right. See, now we're staying higher the entire time, all the time. And then our difference becomes magnified over here. So those animals, so when you select for fecal A count breeding values, you're selecting for healthier animals that generate better immune responses. And if you guys haven't realized this yet, within the next 2 or 3 years, your ability to purchase and have access to antibiotics, coccidiostats, in feed stores is going to be gone. Right? So I'm from California. My folks still raise sheep in Northern California. They, right now on a bottle of LA200, it says restricted drug in California. Coccidiostats say restricted drug in California. You have to have a prescription from a veterinarian. If the veterinarian hasn't been out to your farm in the last 6 months, they cannot write you a prescription. They have to come out and see your facility. So that turns feed stores into pharmacies, right? So that's what's happening, and it's coming to us soon, right? In West Virginia, it'll happen in 2023. So we've got a couple more years. So start finding those veterinarians that are going to help you and start building those veterinary client-patient relationships. Go to your next slide. Yeah, go ahead, next one. All right. So we started thinking about this in response to generalized bacterial disease. And we can model this by using what's on the outside of most gram-negative bacteria, and that's called lipopolysaccharide. So it is a fat and carbohydrate combination. It's a little fat and sugar molecule that's on the outside of a gram-negative bacteria. So we take St. Croix sheep and Suffolk sheep and are starting to study the differences in their responses. So I can take cells, that's why I'm so good at bleeding sheep. We extract blood from the, or cells from the blood, then we can take them in the lab and then expose those cells to LPS, right? So that would be like mock bacterial infection. So in the lab, we can do this. So what happens, just like this thing right here, toll-like receptor 4, that's the receptor for LPS. We are all born with that. You have toll-like receptor 4 on your cells. That starts that response. When LPS binds it, it gets upregulated to produce more toll-like receptor 4. The St. Croix do it, the Suffolks don't. I can keep going this all day long. This little thing here, this is a transcription factor that goes into the nucleus that upregulates these two things. St. Croix do it, Suffolk don't. Come over here, we get this IL-1 beta that is downstream of this. St. Croix do it, Suffolk don't. Come over here, tumor necrosis factor alpha. This is what causes your temperature to go up after you get sick. St. Croix do it, Suffolk don't. Then we can go into the live animal. And this is where it gets really cool. So we set up an experiment this year where we gave sheep LPS. So I can just put it right in their vein. That's why I'm good at that, right? We can put it right in their vein and look at their behavior, what happens to them, what happens to temperature, take a bunch of blood samples on them all throughout the day. Go to your next slide. This one might be a little slow. So just let me explain how we set this up. We had a group of St. Croix wethers. Then I had a group of Katahdins that had an average breeding value for fecal A count of minus 87. That was the average within that group of Katahdins. So I had low Katahdins. Then I had a group of high Katahdins, and their average fecal A count breeding value was plus 288. Then I had a group of Suffolk sheep. So what I'm going to show you in this video, and this is just my phone video of us walking through the barn, are the St. Croix sheep 2 hours after we gave this injection. If you scroll to the bottom of that video, there might be a little tab that pops up. Yeah, just click it. So what we're going to be looking for is just behavior of the animals. And you guys know what sick sheep look like. Those guys look real sick. They're moving around. Their heads are up. These ones start seeing ears down. There's one laying. Oh, look at that one, right? That one. Ugh. Let's go to the next group. Yeah, walking, getting students out of the way. This is the high Katahdin group. They look rough. Wait for it. It gets better. There's your pen of Suffolks. Can you see? You can see the difference of behavior in response to illness. So you got behavioral responses. So what do behavioral responses prime us to do? Treatment, right? So those ones are going to get treated. The Croix are not. They don't need it. And they're able to generate those responses to everything. So hopefully there's something in here that you can take back and learn and think about. You can go to your next slide. None of this is done in a vacuum. And it takes a lot of people to get all of this work done. So I've got a huge crew of students. Unfortunately, all of those kids are gone now. Every single one of them are gone. She's at the CDC. He's at Penn State Extension. He's at postdoc in New Jersey. He's working for a scientific company in California. He's an extension specialist, a small ruminant extension specialist in North Carolina. Reese is out working for American Angus, and Elizabeth is a research technician at the University of Tennessee. I got a whole nother crop coming in now. We only have 2. I used to have 7. So they're down. We get funding from a variety of different organizations, Katahdin Hair Sheep International, the USDA, National Center for Veterinary Parasitology. I've got excellent collaborators at Virginia Tech that help me with all of this stuff. So there's all of this, questions, comments. I'm excited about this stuff. It's what I do. So, yeah. Oh, you can do it at home, right? If you got a microscope, some salt water, and a fecal egg counting slide, it is not rocket science. But I'm going to tell you right now, on my best day when I'm cranking through this, from setup to takedown, I can do 10 an hour. Right. Now, so that's 10 an hour, and that's going through, and I know exactly what I'm looking for, right? You send them to me for $5 a sample, I'll do them for you, and I'll send those data to you within a week. So if you go to the wormx.info, so the American Consortium for Small Ruminant Parasite Control, it's a tough name to get out there, but it's shortened down to WormX. On that web page, there's a ton of information about managing parasites in small ruminants. And on there, there's a link for low-cost fecal egg counts. West Virginia University, Louisiana State, and Texas A&M have all gotten together to have a common cost between all 3 institutions, so it's $5 a sample. So you pay for shipping out to us, we run your samples, and then we email your data back to you. Okay. It's a little bit cheaper than what's going to happen at any vet clinic, too. Okay. Good question. Other stuff? Yes? Sure. And we do this in Katahdins, and it's utilizing the National Sheep Improvement Program and calculating breeding values. So can you tell, there is variation within the St. Croix breed for fecal egg count, but I'm going to tell you that variation is not going to be as high, and that variation will be influenced with sheep that start experiencing some crossbreeding. So if all of a sudden you're killing these Croix lambs and their dressing percents are getting in the mid-50s, and boy, I really like the loin eye area, ribeye area that's coming off these lambs. That's not St. Croix sheep, right? You're getting some influence of some other breeds that are coming into that thing, and you'll start losing parasite resistance within those animals. And you'll be able to find it pretty quick by doing fecal egg counts. But utilizing large-scale genetic evaluation like NSIP, they will do that for you for a fee. It's a little bit complicated, but there's a lot of resources around to help you do that. And you can find, if I want to know information, we have Texel sheep at the university. If I want to know, I could pull out my phone and show you breeding values on my lambs right now and what my sires are, and I can show you a list of all of their lambs and what their breeding values are. That's the power of doing that, right? So once you get into the system doing it and get going, that's how you can find differences within breed. It's tough, right? Yeah. So I try to I'm very fortunate that Virginia Tech maintains a flock of St. Croix sheep for me to use. They use them for some other stuff, but mostly it's for the research that we collaborate on. So they maintain that flock of ewes down there. and then we'll flip a coin about who's buying rams this year or next year. And there's a couple producers around that I buy from in, Kentucky, West Virginia, that we can get rams from that we know they're going to fit what we're trying to do. So no real selection on any phenotype. You look like a St. Croix ram, can you get used bred? You're for us. Yeah, I mean, the St. Croix should be pretty resistant just all by themselves. You shouldn't have to put a whole lot of effort in there. If you got a St. Croix that kills itself with parasites, then that's one less St. Croix that you need around. Yeah. Yeah. Oh, you sent yours to me? Okay, great. Oh, perfect. Criswell, right? Yes. See, I've got a memory for some things. Yeah, you should be leaning pretty hard. It's a tough, and once you get used to the system and understand the quirkiness of the software, it's fine. Did you have something? All we're saying right now is that when you select animals for being more parasite resistant, you're also indirectly selecting for disease resistance or immunity, generalized immunity. I'm a little worried about St. Croix sheep because what can happen is that your immune system can become polarized, where you get really good at responding to one type of thing and really, really bad at responding to something else. And that happens with mouse strains. So we've got mice strains that will respond to parasites really, really well. We've got another one that will respond okay, but they generate the same response, but just not as high. So that can happen. And what I've noticed with some of the Croix that we've run is that they're a little bit more sensitive to coccidia, right? And that makes sense because coccidia has a different type of immune response than the immune response to a helminth or a worm parasite. So coccidia are intracellular. They're not worms. They're these little guys that get into the cell in the small intestine. They replicate asexually, then they replicate sexually, and they burst at 2 levels. So by the time that you see oocysts in the feces, the damage has already been done. Yes. And you start scraping off the lining of the small intestine, you've destroyed that animal's ability to absorb nutrients. So you get some of those lambs that just take forever to, they're 8, 9, 10 months old, and now they're starting to gain. Well, they've just been rebuilding gut tissue for the last 3 or 4 months, and that takes some time to do. So I don't think that just because you're parasite resistant, you're disease resistant. But I think that when we start selecting for those genes in animals that hadn't had that pressure before, we're improving immunity. Can we go too far with it? Do we have a tendency of going too far with it? You bet. So we've got to keep that in thought and moderation. Yep. Yeah. Yes. Sure. All right. So our worm's going to become resistant to the immunity in animals. So when we give an animal, or when we give a drug against a parasite or a bacteria or a virus, it has one point of stopping that particular pathogen. So it's one thing. When I'm up here, so I usually refer to the immune response in St. Croix sheep as the kitchen sink. They're throwing everything at it. So it's not a single prong, and there's redundancy after redundancy. You don't have this, and then you get this. You don't have this, you get this. So yes, natural immunity is much more robust and much more broad spectrum. Now, we can get into all sorts of conversations about what's better, being infected with COVID or getting a vaccine. We can do that once the video's off. But it's one of those things where you get with these drugs, there's just one point. So drugs target a thing, right? And all that thing's going to do is immobilize the worm so the worm can be flushed out. They don't necessarily kill the worms. Like Prohibit doesn't kill worms. Prohibit paralyzes larvae, and then they leave the system because they can't swim. They can't swim, they can't maintain position in the gut. Yes?
So when you do your first initial inoculation of worms,
Mm-hmm.
and then you said you gave them all dewormer
Yes.
package,
Yeah.
what was the deworming protocol?
I only use Prohibit. That's it. And from a scientific standpoint, there's a rationale. Prohibit's out of their system in 72 hours. I don't want to have persistent effect. So I don't use macrolytic lactones or Sidectin, Dectomax, ivermectin. I don't use any of those. I don't use Valvazin. Valvazin in most sheep doesn't work anymore. Works great in cattle. All the cattle producers have gotten away from white dewormers because porons are a lot easier than walking around the front side of a chute. So what happens in cattle now is that we're seeing a lot of resistance to poron dewormers. Sidectin, Dectomax, Eprinex aren't as effective anymore. So that's what I do. When I do I run our goat test at West Virginia, so we had 172 Kikos that came in from 16 different states and 39 different producers. They got 3 dewormers when they showed up on the first day. They got Prohibit, they got Sidectin, and they got Valvazin. Because there is synergistic effects between the drugs. They don't go into the same tube. They get 3 different drenches. And all the work by Dr. Kaplan will tell you that that's the more effective approach. Right. Right. So I use 3 because I've got goats coming in from as far west as Kansas, Mississippi, Alabama, Florida, Georgia. I've got all the Gulf states that are sending me sheep or sending me goats. I don't trust any one of them. So, and I don't know what their worms are resistant to on their farms. All I know is this last year we had a 93% fecal egg count reduction by using the triple cocktail dewormer. So we don't put it into one thing. It's important to remember we use 3 different syringes.
And you don't have issues with any of them not tolerating the Prohibit level?
So I get Prohibit from a pharmacist in Oklahoma. So I don't buy packaged Prohibit from feed stores. And the reason I do this, it works a little bit better than the packaged Prohibit. I use a lot of Prohibit. So my first purchase as a faculty member at WVU was a kilo of Prohibit from a pharmacy in Oklahoma with my P card. So I didn't get any trouble, but I raised a lot of eyebrows, right? What are you doing with this P card? So I buy a kilo at a time from Milt Simpson, and he runs a pharmacy in Oklahoma. And he's worked with veterinarians and researchers in that area. And I get a little tub, and it's a light block tub, and he sends it to me. And then I can weigh out how much I need because I've got scales, and I got scales, everything, all the stuff to mix things. It is so much easier to mix. You've read the back of those Prohibit packages. It makes you kind of cross-eyed and crazy and like you.
What I was always taught is you do like the 2 dewormers. For my goats, my go-to is like the Sidectin and Valvazin.
Okay.
They work really good for us. We've gotten down to really, the majority of the time we only deworm after kitting.
Okay.
They have a couple of those that we have to do one more time.
Well, that makes sense to use Sidectin in that case because that's going to be more effective on haplobiotic larvae that may still be there, right? So you've got some larvae that are coming out of hypobiosis, and it's going to be effective on that. L4 stage is kind of this really weird, because when they go into hypobiosis, they're not like full-on worms that are going to say, I'm going to freeze. It's the new L3s that are coming in saying, I'm going to stop development. And I'm stopping at this point, and then once conditions are right, I'm gonna start growing. And the L4, even the St. Croix are blind to the L4. They don't generate an immune response to L4s at all. At least that's our data, and what we've published is that they just can't see L4s with their immune response. So I've got one of those experiments in my back pocket where I'd love to cannulate a St. Croix sheep and then put adult worms right into their abomasum to see if they generate a response to adults, because they really never have to deal with adults. Right. Or the adults that are there are at such a low level, it doesn't impact or doesn't need to generate a big immune response. Oh, yeah, they're out. Yep. So I'm always been kind of curious, man, if I can kill a St. Croix, not kill, if I can get a St. Croix sheep to get sick with worms by giving it adult parasites. Yeah.
Well, we've always been told or taught that prohibit was like your last ditch effort. Like if other things weren't working, like then go to prohibit. We did have a doe one time that we used prohibit on. Had the scales out, weighed my doe, measured everything out appropriately, and she almost broke on me.
Uh-huh.
Like 10 seconds after the prohibit hit her, she was down and stopped breathing.
Go easy on your goat recommendations on the prohibit dosing. Prohibit is toxic if you give it at too high of a level. And we sometimes have a tendency to give them a little bit more than what they need, because we work in this area where a little bit's good, more's better. So we just have to be careful with that part of it. I use prohibit simply from a scientific standpoint. That is, I know there is zero left over in their system that's going to impact the infectivity of when I infect them again. If I use Sidectin, I got to wait 21 days in between there. If I use prohibit, it's gone. I mean, it's gone in 72 hours. So that's the difference. Yes.
Once you have a sheep that's dead,
Uh-huh. Yep.
what's the recovery?
There's so many things that affect that. Age of the animal, their body condition going into that, how much mass do they have to give up a little bit? So if you've got a really thin animal that is sick with probably multiple things, and it's not just parasites, recovery rate's going to be trash, right? If you've got one of these real moderate to high body condition, like 3-4 body condition, she's in good shape, she's just coming in and weak, they're usually fairly recoverable. So there's a lot of things that affect that. B vitamins, iron, because the animals, it takes a long time to recover red blood cells. Red blood cells don't just get replaced like this. It's about 2 weeks to develop a new red blood cell. So even though that process is constantly going, if those worms are bleeding the animals, I've had rams get bled out from the inside out. So we paid way too much for the Suffolk ram. He came out to the farm at the university. We did everything. Dewormed, do all the stuff, get him ready to breed use, put him out there. 5 days later, he's dead. Laying up against the fence, dead. Straight up, feet straight up. He had not experienced rigor. He had been dead for about 8 hours. And he was still loose. Right? So we open him up and we're like, well, okay. So the vet's out there and I look at her and I said, where's this animal's blood? All of its muscle is pale, heart, liver, lungs pale, spleen's pale, kidneys pale. And I said, you know what? I said, this is probably some kind of crazy metabolic thing, right? And I said, let's just open up the gut and see what's happening. That's where the blood was. There were so many worms in that animal's abomasum, it looked like a shag carpet inside the abomasum. And it was a great thing because my students at the time had let our larval culture dissipate, so we didn't have as many worms around. So we spent the rest of the day collecting everything out of that one's abomasum and crushing up the adult females to get their eggs, and then culturing their eggs and the larvae and doing that stuff. So I don't like to give a whole lot of treatment recommendations and those kind of things because I'm not a veterinarian. I just know what kind of things are going to affect recovery rate.
So knowing what you know about the Croix, Katahdin, and bull breeds, what do you think breeders of St. Croix should be focusing on?
The St. Croix breed does one thing really, really well, and that in a parasite challenged environment, your sheep survive where others do not. Right? You could grow your sheep on pasture in Arkansas in the summertime, and they would live. The Katahdins would die. Right? That's you guys in Arkansas? Okay. So you know Joan Burke down there and at the USDA station that runs the Katahdins over there in Boonville? Okay. Okay. So she runs Katahdins in Boonville in Arkansas, and just it's tough putting them on grass in summertime because even the Katahdins will keel over. So you can grow sheep in a parasite dense environment. You cannot expect your sheep to compete with other breeds for muscling, right? But your sheep will breed out of season. They will have their good mamas. They're not particularly good milkers, but their lambs don't need as much milk, right? So there's some trade-offs with this thing. So it makes you start to think that you need a terminal sire just as bad as Katahdins do. And that's where I come in because I'm a Texel breeder. And that's what has really kind of taken over the Katahdins and even the Texel groups. We sell a lot of rams to Katahdin producers because the Texels have parasite resistance. We've done studies on the mechanism in the Texels, and it tends to be more focused to the adult worm than the larvae. So we've got some data on that to help out with those type of things too. So what's the best thing that you can do? You keep marketing your breed as the model of parasite resistance. You are the epitome of parasite resistance. That's your claim to fame. And be careful about anything else, right? Because you're not going to convince me that they are super heavy muscle. Ever, 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 ever. Not when I've got lambs that scan with a 4-inch ribeye area at 70 pounds on my Texel lambs. You're not going to convince me that a Croix is ever going to be capable. And honest to God, I don't want a Croix to do that. I want a Croix to survive on pasture with limited input. They are a low input maintenance breed, maintenance ewe breed, that we should be using terminal sires on to generate crossbred progeny. That's it. Right? Dorpers are fine. They've got no resistance whatsoever. They were developed in the desert in Africa. Right? Okay, so they grew up same place that I grew up. My wife didn't understand. She didn't understand the concept that I would go hunting out of my backyard. We just had jackrabbits everywhere in the desert. And that's when we got home from school because there was nobody else's house anywhere around us until she got them out there. She's from Greensboro, North Carolina. So she comes out to the desert. She's like, oh my God, I understand now why there's nothing else to do. I said, we used to put pennies on the railroad track and watch the trains go by, right? We would go shoot rabbits at the alfalfa fields that are out there. That's what we did. So that's the same kind of environment, a very Mediterranean, dry environment. They have no resistance, and there's no reason to expect them to have any resistance to that. So just because they have hair doesn't mean that they're resistant.
Right. And I guess we've gotten fortunate because our Holt has done pretty well in our winter.
Right.
Right, he pretty well stays by himself unless he doesn't get exposure to a lot of the
Mm-hmm.
But I know you said when you start crossing with that same core, you start decreasing that immune response. Have you done studies in like those F1 first generation crosses? We've never kept any, but we have decided to keep a few ewes back.
There's
We
a
actually brought a couple with us today.
there's a lot of people out west in Oregon, California that really like the Dorp Croix. They love Dorper St. Croix crosses, and it's a really great cross if you've got the environment to support it. If you've got a highly parasitized environment, those crossbred animals are going to be okay, would be my expectation, right? but you've got a multitude of terminal sire options. If you want to keep them hair and keep those lambs with hair on them, and if you are getting a premium for selling lambs with tail and hair on them for different ethnic markets, by all means do that. But if you cross with the Texel, you can go to my website and see what it looks like when a Katahdin gets crossed with the Texel versus a Suffolk. And there's a ton of carcass images on all of that stuff. We've got birth weight, all the maternal stuff on the Katahdin side of what happens with that. So there's a ton of options for terminal sires. And I'm not saying that Dorper's a bad option. It's just not the first one that I would think about in this area. There's some stuff, well, a lot of that stuff died after Dr. Parker stopped doing research at Ohio State. And there wasn't a whole lot of activity with St. Croix sheep until, the mid-90s, and then it stopped, and then we started picking it up again in late 2000s, and then started doing more stuff with them. And we haven't really been looking at breeding the St. Croix, doing anything, because there's no need to. The Katahdin stepped into that role, right, as doing that. And the St. Croix is a progenitor of the Katahdin. The Katahdin's a little bit better on those kind of things, and bigger birth weights, bigger weaning weights on the Katahdins. And they've just kind of taken off in popularity. So I don't think that it's a bad idea. I think that you can go through some of the older literature and take a look at that. And if you want to talk with me about it, we can go. I can spend some time looking up those kind of things. I just don't know them off the top of my head. I just know the guys out west, some of those Dorp Croix ewes work really well in intensively managed range situations where you don't want to worry about shearing. So, and there's a lot of Dorpers out there too. I mean, the guys in Texas and California love some Dorpers, but they also don't deal with a ton of parasites either. Yeah, right. Uh-huh. There are some of these breeds are collecting fecal A count data. So you can find Suffolk rams with fecal A count breeding values. You can find Texel rams with fecal A count breeding values. You can find some Dorpers with fecal A count breeding values in Texas. So I think that there's options for you to do some crossbreeding that extend past the Texel or those kind of things, and can stay with some hair sheep. There's some Dorper breeders out in Texas that will have some parasite. Well, they'll have some estimated breeding values for that too. And there's even some producers in Tennessee that do. So that's, if I was going to find, I mean, your biggest problem is if you want to try to run lambs on grass in the summertime, they're going to die if you start using a lot of the other breeds. Your parasite load is so high that if they don't. Right. No. You don't want to lose lambs to something stupid. So your thing is that you've got low input lambs that are going to survive on pasture where everybody else is going to have to feedlot them. You're going to take a hit when you go to the sale barn a little bit because of muscling and those kind of things and size of the animals. But honest to God, you should be marketing your animals to a completely different group, right? And you should not be competing with this, like that sale in Mount Hope, Ohio, or any of these big sales around there, because they're graded auctions that are getting sold or split out by muscling. They see your sheep coming through there, and then they see my trailer full of cull Texel lambs that are just walking around with big butts and big shoulders and everything else. You're going to get hammered a little bit on that. But your cost per pound of production is so much cheaper than what I'm paying, because I'm buying $8 a bushel corn. Yeah, right. Well, it's a Mustang versus a Vespa, right? How about that? Maybe that. Um, you know, I'm going to be able to get a little bit further faster. You're going to be able to get that same distance. It's going to take you a little slower, but you're going to pay less to do it. Yep. hmm Well, St. Croix are going to have more concentration of the genetics for parasite resistance than a Katahdin will. hmm A little bit, right? But it's closer genetically, right, to that Suffolk. I mean, it's just happened sooner in their pedigree. So that happened a long time ago with Michael Peel in Maine. So it's been a while. And people have put Suffolk in. I mean, Katahdin's got an open registry. So you can still register 87.5% sheep. And you don't have to say what the other 12.5% was. So. Mm-hmm. Yep. Mm-hmm. Well, what's your U base? What's the group of U's that you're using? And then you're using Katahdin as a terminal sire on the St. Croix. Okay, so the St. Croix U doesn't handle a big lamb well. And I'll give you my example for this. So we had those St. Croix U's down at Tech. Dr. Greiner had brought in, he was doing some work with muscling on sheep and relative to frame size. So he had bought this really fast growing, big framed, heavy muscled sheep out of Michigan that was a monster, right? He had a 4-inch eye. He also had one that was half his size that had a 4-inch eye. So they were doing some of that work. That big Michigan ram got in with those St. Croix ewes. I had a Croix ewe with 30 pounds of lamb in her. That ewe doesn't have enough room to eat enough feed to support the growth of those crossbred lambs. So you got to be real careful about what you're expecting that ewe base to generate beyond purebred St. Croix sheep. Moderate to small to moderate, so you're controlling your birth rate, your birth weights. I don't either. There you go. Right. Yep. Yep. You can get the genetics from the ram side of it, and don't have to. Then that'll open up those ewes a little bit to more flexibility, so that. But your ewe base, right, is what you're managing year round, and that's the one that you have to think of that needs to be low input, easy keeping, is going to survive well in your area. And it's just like cattle. I mean, the cattle that work in Arkansas are not the cattle that are going to work in West Virginia, right? Okay, so we don't run a lot of eared cattle in our neck of the woods. No. We've got frame 8 Angus that are walking around all day because we've got grass for days in those hills, right? I want to kill these guys. I mean, yeah. And in Northern California, we've got, we're running frame size 5 cows because there is no, I mean, they're grazing whatever the fire service didn't take out or whatever the fire didn't take out. My judging team coach lost all of his cattle on a fire that blew up 90,000 acres in there. So different cattle, different environments, same thing with sheep, right? Different sheep for different environments. Your environment, you're going to need a easy doing you. So those Dorper Croix, the Katahdin Croix, that's what I might even be thinking about more, is just some Katahdin Croix. Right. And then on those crossbred ewes, you just bring in some terminal sires without getting too big on them and making sure that all your lambing is happening as low input as possible. Yep. It's been fun. Okay. What else you guys got? depends on where you live. So for you guys, yeah, you'll have egg counts February, March. Wouldn't surprise me if you had a little bit of egg count in January, but it's going to be different species. So you're going to find homuncus happening when it's starting to get hotter outside. You're gonna have, but I can't tell the difference between a homuncus egg and Teler drossagia or Ostraced or Trichostrongylus. They all have the same egg shape, so I can't differentiate those eggs. I have to grow them out to larvae and then measure the length of their tail to see any differences. Yep. Okay. Okay. Right. Keep the suffix in Colorado. Right. And you got to shear them, right? Mm-hmm. Yeah. Mm-hmm. Mm-hmm. So it sounds like you're on the right track. You've applied what you know from the cattle world to sheep and done so in a thoughtful manner. That's all right. Yeah.
What's your next step as far as research?
Well, this is our latest publication. So that's a worm. It's an adult worm, and those are cells on the outside of its head. And then the cells have released their DNA, and that makes that cord. That's just DNA in that cord. So that's electron microscopy. So I get to do like on the barn sheep work and this stuff, which is pretty fun. We're really going after kind of understanding immune mechanisms and multiple things in the animal. So that's what we're doing the LPS stuff now. So I've got a couple grad students working on that. I've got another one looking at effects of parasitism on feed efficiency and parasite resistance and how parasite infection changes feeding behaviors in animals. So we've got a group of Suffolk sheep in a grow safe barn. We can measure intake on a daily basis into those animals. So they're pin-fed animals, and they've got an RFID tag. They stick their head in the trough, and it measures how much feed they remove. It measures how many times they go there every day. It records all those data. So he's got 2 or 3 years of doing those experiments, and he just got hired as he left his PhD early to take this job in Pennsylvania. They had a county-level agent position open that was sheep-specific, and those don't happen anywhere, right? So I told him to apply for it. He got that job, and then uh-huh. Yeah, how you translate those information real time is complicated because this feeding system, each one of these nodes costs $10,000, right? And then you have all the technology and everything else, and you got them in a barn and you're feeding them that way. So shipping's free on Amazon Prime, but not installation. So that's the tricky part with that. So what we're thinking about is how this affects that initial behavior and how that may exacerbate. So can you change the feed type that's going into the animals so that the little amount that they're eating is actually helping them more? Does that make sense? Yep. That's all of those questions. We've got he's got pages and pages of data to ask all of those questions of those data because we have everything. There's 40 lambs in that barn. they had a primary infection, they were cleared, they got a secondary or a challenge infection. So we know what happens during the first one, we know what happens during the second one, how that changes eating behavior, growth, everything. Fecal A counts, all of that stuff. Yeah, and we've got a group of animals that weren't infected and ones that were. So we can see the differences between them. I got another, how many more years? 30 years? Because I started late in life, so. So I thought about retiring at 60 or 62 and probably gonna be like 75, so. You know, I have a couple years of retirement and then, yeah, that's right, that's right. Okay. Yep, yep. Well, feeding behavior, that's the only thing that we can pick up on is how. Oh, yeah. I mean, how many times they come to the trough? How long do they stay there? We know how long they've got their head in there because it marks it when the ear tag goes in and when it comes out. How much feed did they eat while they were in there? There's tons of, yeah. Yeah, yeah. It's just some of the stuff is, it just takes a lot of analysis. Right. Correct. Mm-hmm. Yep. This recording's